Let's move to the next topic. Intergenic gene interactions. Intergenic gene interactions. What's meant by intergenic gene interactions? Intergenic gene interactions is the interaction between the alleles at different loci. What's the definition? First learn the definition. What is intergenic interaction? The question is intergenic gene interaction. Definition: the intergenic gene interaction is the interaction between the alleles at different loci. So first you have to learn what is allele and what is loci. Alleles and loci. This is the homologous chromosome. You all know there is a homologous chromosome which derive from paternal and maternal, a male and a female. The chromosomes which derive from paternal and maternal. This is derived from male and this is derived from female, father and mother. So paternal and maternal. This is called a homologous chromosome. In the homologous chromosome, these are the alleles. For example, here small r, p, p. So these are alleles. The alleles are, what are alleles? The alleles are present in a pair. So these alleles are located in different locus. So this is called the locus. This is one locus, this is another locus. So a pair of genes are called alleles. So what is intergenic gene interaction? It's the interaction between the alleles at different loci. There is an interaction between these alleles at different locus. This is one locus, this is another locus. So interaction takes place between these alleles of different locus. From this locus and this locus, interactions taking place. That's called intergenic interaction. So here there are some examples you are going to study. Intergenic interaction, which is otherwise called as non-allelic interaction. What's the other name of intergenic interaction? Non-allelic interaction. So in this interaction, there is an example, epistatic interaction. There are number of epistatic interaction which gives in different ratios. What are the types? These all are the different types of epistatic interaction. So today you are going to concentrate only one type. Before that, just you learn what are the different types of epistatic interactions. Number one, dominant epistatic interaction. First epistatic interaction is dominant epistatic interaction. Then recessive epistatic interaction. Duplicate gene with cumulative effect. Complementary gene interaction. Supplementary gene interaction. Inhibitor gene interaction and duplicate gene interaction. This all gives different ratios with different examples. For 1 word question, 2 mark question as well as the 3 mark question, these types of epistatic interactions are important. What are the types of interaction? What's the example of this and what is the ratio? Let's see one by one. First one is dominant epistatic interaction. Dominant epistatic interaction is observed in fruit color of summer squash, cucurbita, pepper, summer squash. In summer squash, it gives the ratio 12:3:1. So this is 1 word question. 12:3:1 is the ratio of dominant gene epistasis. Recessive gene epistasis. Epistatic interaction. Recessive epistatic interaction. Example, flower color of antirrhinum. So here the example is antirrhinum, which gives the ratio 9:3:4. 9:3:4. Then duplicate gene with cumulative effect. So first one is dominant epistatic, then recessive epistatic, dominant epistatic which gives 12:3:1, recessive epistasis it gives the ratio 9:3:4, and duplicate gene with cumulative effect. The example is fruit shape. Here in dominant epistasis the color of the fruit, and here is the shape of the fruit in summer squash. Both the examples are summer squash, but in dominant epistasis we are consider the color of the fruit in summer squash, but here the shape of the fruit in summer squash. So here it gives the ratio, it's duplicate gene with cumulative effect. So it gives 9:6:1 ratio, 9:6:1. Then complementary gene and supplementary gene interaction. Complementary gene interaction, the example is flower color of sweet pea. Flower color of sweet pea. So it gives the ratio 9:7. 9:7. And supplementary gene, the example is grain color in maize, which gives the ratio 9:3:4. And inhibitor gene, the leaf color in rice, leaf color of rice which gives 13:3. And duplicate gene epistatic interaction which gives 15:1 ratio. The example is seed capsule shape. Seed capsule shape. The ratio it gives is 15:1. So these all are different intergenic interaction which is otherwise called as non-allelic interactions. So among all we are going to learn only one example today, that's dominant epistatic interaction. Let's see the example. Before that, what is dominant epistatic interaction? Dominant epistasis. What's meant by dominant epistasis? The two alleles of a gene at one locus interfere. So again you look at this diagram. The two alleles here, two alleles are present in different locus. The two alleles of a gene. So this is allele. Among this allele there is a gene. This is one gene, this is one gene, which get together as a pair. So this pair of genes are known as alleles. So what is dominant epistasis? The two alleles, this is one allele, this is another allele. Two alleles of a gene at one locus interfere and suppresses. Interfere means it prevents the continuity. So it prevents, it interferes. So one locus, two alleles of a gene at one locus interfere and suppress. Suppress means it mask or it hide. So this one locus genes which suppress the phenotypic expression of the different pair of allele of another gene at another locus. So this pair of gene, that is allele, in one locus suppress or mask another pair of allele in its phenotypic expression. So different pair of alleles of another gene is suppressed by another locus. This is called dominant epistasis. So let's see the example in summer squash. Summer squash, we are taking the example as the color of the fruit. The color of the fruit. Here we are taking the example, the parent is here, it gives the ratio 12:3:1. So in dominant epistasis, which gives the ratio 12:3:1. So in this, there are two terms you have to learn. One is epistatic and hypostatic. Epistatic and hypostatic. What's meant by epistatic and what's hypostatic? Epistatic is the gene that masks the phenotype. As I mentioned earlier, here two alleles with different locus. one locus of allele which mask or suppress the another locus. So, what is epistatic? The gene that masks the phenotypic expression of the another locus. If this gene, this allele of this locus masks this character, this is called as epistatic. What's the definition? The gene that masks phenotypic, this gene masks the phenotypic ratio, phenotypic expression of another locus. So, this is called epistatic. And what's meant by hypostatic? The gene whose expression is interfered. So, this one masks, and this one, the gene expression is interfered by non-allelic genes. So, which prevents, this character is prevented. So, this is known as hypostatic. So, first learn the definition. What is epistatic? The gene that masks the phenotypic expression of another locus is called epistatic. What's meant by hypostatic? The gene whose expression is interfered by non-allelic genes and prevent from exhibiting. So, it never exhibited because this locus allele, which the genes present in this allele of this locus mask the character, that is epistatic, and this expression do not exhibiting, that is called hypostatic. I hope you understand it. Now, let us see that dominant epistatic. What's the example? Here the parent taking as fruit color, the character is color of summer, fruit color of summer squash. In summer squash, we are taking the parent. There are white color flower, white color, make a cross between yellow fruit, white fruit with yellow fruit. So, here we are taking the white fruit color is white fruit. Here this is the fruit, white fruit. In this white fruit with yellow fruit. This is the yellow fruit. So, in yellow fruit, this is the color of yellow fruit. So, white and yellow. So, the gene pair, the allelic expression is capital W, small g with small w, capital G, capital. This is the parent. The parental character is capital W, the white fruit and yellow fruit. So, capital W, small g and small w, capital G. This is the parent. And what are the gametes? From here, take out the gamete. Here, capital W, small g. This is the gamete. Here, small w, capital G is the gamete. So, when you make a cross between these two, in F1 generation, it gives a heterozygous hybrid. So, these are parent white color fruit and this is yellow color fruit. When you make a cross between these two, it gives capital W, small w, capital G, small g. So, capital W, small w, capital G and small g. Now, this is heterozygous in the F1 generation. Now, normally in the F1 generation has to cross. So, hybrid with hybrid, this one with capital W, capital G, small g, hybrid with hybrid. So, it gives the ratio 12:3:1. Let us draw the checkerboard and find out the ratio. Here it produces 16 different offsprings. 1, here 4. This is the column. You have drawn this. And take out the gametes from here. Here what are the possible gametes? Capital W, small, here capital W, capital G, capital W, small g, small w, capital G, small w, small g. So, capital W, capital G, capital W, small g, small w, capital G, small w, small g. The same, here one set we can take it as a male, another set take it as a female. So, here it gives capital W, capital G, capital W, small g, small w, capital G, small w, small g. So, fill it and it gives the ratio 12:3:1 in this character. It shows the dominant epistasis. So, capital W, just write it, capital W, capital W, capital G, capital G. Here capital W, capital W, capital G, small g. Here capital W, small w, capital G, capital G, capital W, small w, capital G, small g. Then this and this. Always we can write the capital letters first. So, dominant character, capital W, capital W, capital G, small g, capital W, small g, small g. Here, capital W, small w, capital G, small g, capital W, small g, small g. Here, capital W, small w, capital G, capital G, capital W, small w, capital G, small g, small w, capital G, capital G, small w, capital G, small g. Then this and this, capital W, small w, capital G, small g. Here, capital W, small g, small g, small w, capital G, small g, and small w, small g, small g. So, here the dominant character is W, white color. White color, it gives this capital W, wherever the capital W is present, it gives the white character, dominant character. Wherever the small w is present, small w, which gives the yellow fruit. And wherever the homozygous recessive is there, which produces the green fruit. So, 3 different ratios it gives in the ratio 12:3:1 as the dominant epistatic. So, here find out the color. This is white. This capital W is present, so it gives white. Here capital W, so it is also white. Wherever the capital W is present, it gives white. White. Here also white. Capital W, so white. Here capital W, white. The fruit color is white. Here the fruit color is white. Here the fruit color, white color. Here also white. Here small w is present. Wherever this small w is present, it gives yellow. The fruit color is yellow. Here also small w, so it gives yellow color. And here it gives capital W, so wherever capital W is there, it gives white. Here capital W, so white. Here small w, so it gives yellow. And finally, this small w, small g, which gives the green color. So, this is the one, it gives green. So, 12:3:1. Let us check the ratio. Here, how many white it gives in the ratio, that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 white fruit color is to 3 yellow. 1, 2, 3. 3 yellow is to 1 green. So, this is the ratio. It gives that dominant epistasis. The ratio is 12:3:1. So this is the example of dominant epistasis. So dominant epistasis. So today you are going to learn only one question, that is dominant epistasis, dominant epistatic interaction. The example is fruit color in summer squash. So example of dominant epistasis is summer squash or cucurbita pepo. So in summer squash, it gives the ratio 12:3:1. So what is dominant epistasis? Two alleles. So in this case, here wherever the capital W is present, the dominant character, this white color, mask or suppress any other color. It masks the yellow color or the green color. So colored fruit is masked or suppressed by this white color because of its dominant nature, it masks. So this is epistatic, and this small w and small g, that is small g and w, small w is hypostatic, because epistatic and hypostatic. What is epistatic? The gene that masks the phenotypic expression is called as epistatic gene, and hypostatic is the gene whose expression is interfered. What's interfered? It's prevent from continuing. That is hypostatic. So here you learn three definitions. So what is intergenic interaction? Interactions between the alleles at different loci. Interactions between the alleles at different loci. You understand clear what are alleles and what is the locus? Within a homologous chromosome, there are locus, different locus which contains genes present in the form of alleles. So these are individual genes. At each genetic locus, individuals has two alleles. So individuals has two alleles which are located in different locus. And dominant epistasis, what are dominant epistasis? The two alleles of a gene at one locus interfere and suppress or mask the expression of the different pair of allelic forms of another gene in the another locus. Now then there are some examples. Epistatic interactions, it shows different examples, dominant different types with different examples in different ratios. Dominant epistasis, recessive epistasis, duplicate gene with cumulative effect, complementary gene interaction, supplementary gene interaction, inhibitor gene interaction, and duplicate interaction. What are the different ratios? Dominant gene interaction ratio is 12:3:1. Recessive epistasis 9:3:4. Duplicate gene with cumulative effect is 9:6:1. Complementary gene ratio is 9:7, and supplementary gene is 9:3:4, and inhibitor gene is 13:3, and duplicate gene is 15:1. What are the examples? Dominant epistasis, the fruit color of summer squash. Recessive epistasis, the flower color in antirrhinum. And duplicate gene with a cumulative effect, fruit shape of summer squash, shape of the fruit in summer squash. Then complementary gene, flower color of sweet pea. Supplementary gene, grain color in maize. Inhibitor gene, the leaf color, rice, in rice plant the color of the leaf. And duplicate gene example, seed, capsule, shape of the seed capsule. So before the next class, learn all this question. Thank you.